Hello students, welcome back to the CMCD tutorial channel. Biology chapter 10 Microbes in Human Welfare. Chapter 9 Akizauta chapter 9 and Khalautin bangkat previous classes ya in bangkat event deujin chapter 9 Akizauta ma to chapter 10 Microbes in Human Welfare. To microbes is a emu thelo microorganisms je hiya male microbes is microorganisms atampi akha mihing le singte ai lo le ganing tia dinga kha microbes je kha na le hon tung tu le hi a hoi lo na ti na tuam a malaria chi khoma mosquito as a care vector malaria chi khoma hi elephantiasis ringworm flu typhoid common cold chi khong le it can cause, all right, but then in this chapter just throw out all those negative effects of microbes. We are going to study how it helps the human welfare. A patop na dinga ka banga litinga hinna litinga ligidona ha hon pan pi white ka house human welfare kichi. So they are protozoa, bacteria, fungi, virus of plants and animals. Mom. And then ama sapina ka na insunga ina ibol theo household products sa ka za thele tam pio mention ai kha en masale mi a household la is ochanga kha a industrial mo industry as a whole alian zo bangkat zu ji khong ma aba na sewage treatment ji changa kha tuinin te suksian na aba na biogas bang ji form di biogas ji changa gas ta na ai silit wang kada gas ji dan wa kha an me wan na ki zang thema hoi ka microbes tin bang ji dan do pan pi thewa biogas form the dinga mo microbes jen aba biocontrol agent sa le ki zang di biocontrol ji changa chu to mosquitoes te thogang te khong ha bang ji dan de wa that thewa living organisms ma zanga chemical sang lo amo as a biocontrol agents. How can we kill those insects which can cause diseases to the plants as well as humans? About now how they can be used as biofertilizers, nitrogen fixation quate microbes in panpiam chi. I think the high chapter mo tamsim 3, 4, 5, 6 topics ta kahia microbes in human welfare. Masapian na microbes in household products. Microbes te kha diversity le an mo om now at tui sada sale hingzawom avot avodale hingzawom male al namonale omzawom avot na satna alu. So everywhere they were found, everywhere, literally everywhere, all right, in soil, water, air, inside our bodies igil sungale aho ileom aho ilole mo. So maka microscopic midamu theilaw hungchin a diversity theitamo. To next household products, how they can help in household products. Household products are hatun elle card na zangai diwama dahi ji homa card dahi ji khong ai lo le cheese ji khong here do ji khong bread khong bol na mo puri ata bakhat ata paratha waya kha do ki chia bana cheese ji khong hai ti khabang chidan dewa ki bola microbes te sangamo. We are going to study what are the microbes involved in these household products. Ama sapena kha bacteria panneula bacteria ji khado mention there is one kind of bacteria, a group of bacteria which is known as lactic acid bacteria. This is a group of bacteria. Haya example lactobacillus, ji khado mention wai pen kha card kha bazar ko mamo avui khong kha chitani hoi kha starter akizang chin inoculum kichi inoculum chubang to start the process male starter means to start the process. You put inside the milk, you buy a milk atuite mo avuite samlo milk and then after overnight they will convert it into card. Dahi male so hai bacteria khabangai chile lactobacillus. It converts the milk into a milk into card. Lactobacillus will convert the milk into card. So how does it work exactly? Milk in protein protein casein they have proteins casein and all those. So those proteins, what happens is that when we put the starter in the column, it coagulation process. So mo sukhalonjin why sukhal na penaha in that process the acid lactobacillus acid acid bacteria here. So it converts that milk into an acidic form. So now it becomes a little bit sweet sour. It's because of the presence of lactic acid. And the name of the bacteria involved is lactobacillus. Important because card it increases the nutritional quality, or it improves the nutritional quality of vitamin B12. Vitamin B12 may more karin so stomach bang stomach card annex so chang I don't chang hilole singpi donzo puri donzo khong I don't chang bang dan de wa ya digestion are important jumale digestion bang jaga how does it help it chang bacteria, there are bad bacteria also in our stomach, alright? Those bacteria, they will damage it. Again, the lactic acid, the lactobacillus will damage them, and then bad bacteria they have engine. So in the digestion process, they were smoothing engine. Vitamin B12 and is yaguamo. Ki don't tank pi. So anexo, sinki don't zonga. Next time you go, alright, to a restaurant or hotel, then you can drink this. So it's very helpful for. And then iron lay increase again mom iron sisan hamale iron sisan tamna ding abana nervous system khong ale hoido pan pi mo nerve cells te khong le pan pi bang khat bol hoi sak so kar kha le don di ge mo pot tinga kha vot jak bangin on le lim lo ma uchi dan de ma kha to kha do ka chi mo do pe na kha do sa li chi khong bacteria I maida ki leita tuito ki hel hoya kha a products ti kha dosa idli hai dia Indian malaya South Indian foods te bang kha North India South India ko ngai chang ma kha I maida chawale zanga to hotel kho pe le maida chaw zanga thil tom to abol the da no ma to up tom a khum hile ali le so chiram ne wa kha dosa idli ti khong kai cha paratha lian dan de ma paratha lian pi khong tom khong bol theu do bang khada para do para ki bol chi hai kha it is nothing but it reuses bacteria ma bacteria kha to puff up chini sulian sak ma hon puak sak hon thathau sak chimene thathau sak puak sak engine why puff up appearance bin khapia mom puff up appearance the puff up appearance of dough, okay, of all types of the food items where we use dough, it is because of due to the production of carbon dioxide gas. So haki mixture kha asunga kha carbon dioxide gas zo sia kha hon por khai jo por khat ngai mo hon por khat changa kha ma kha ya food items chi kha hon puff up chi ge hon po khiao ma and since that is because of the bacteria abana bread ale ki bol po bakery pa idole bakery bread in tenga bread to items items in dele bakers isa para ki bol bakers chu bakers changa chu baking products ei jaka mo yeast engineer yeast pin kha chu fungi mo hai pin fungi baker yeast changa le fermentation ki bolen chin hoi baker yeast pin kha banga min Saccharomyces cerevisiae ki chi spelling kha hoi de wen enula kel khia lo di mo exam changa Saccharomyces cerevisiae Saccharomyces cerevisiae zanga kha fermentation jaga kha puak sak the mo hoi fermentation process kha puak sak the chi nai chu thatho sak the thil him do hilele zui bol janga le zule fermentation thodi hilele azat na tom mo hai kha enchena ba na South India la male to di ki children khado min jin hai kha palm tree mo in palm tree they dig out the soft sap from the palm tree and they make a drink which is known as toddy mo toddy South Indian traditional drink ei local ale baka zu kha chi khong le ki bozil thobai mo chi dan jo kha local la kha village la khonga kha cha adon sek zelu kha todi ki chi that is also done by fermentation of the fermenting the sap from the palms when they ferment the sap from the palms male asia ba para kila abana alas na jinga kha white chow lo le tu ngari chi khong le fermentation yana mo soybean bekan thu ngathu ngatwai thu mo whiting and mai lo mai du mo la yeng tian so all those were also done by fermentation of the microbes mo alas na cheese kha Swiss cheese so mention Swiss cheese pin kha bang bacteria ki zanga ki bolia michile they use a bacteria known as so kachi ma banga mo hai chapter kha species ni abang khat bacteria fungi name te kha tam pi na by hard do mention khat ve thua chu hak samei the le sim leo chin mo used to de sim mun changa chu thile mi sim mo le ni ve thum ve sim to khat ve sim ki bang lo hi mai chu mo le i thei bang lo cha chu mit singa le ge thei ta mo le min teu propiani bacterium sermani one more question ang kidong the switches bona bacteria ki sang pin khabangai chile we use propiani bacteria sermani which is used in Swiss cheese more Swiss changa su Switzerland melo more Swiss watches chi khong na sang le Rolex Swiss watches te khaman tam malam quality le ho inchin in abana ai Swiss cheese kha why Swiss cheese kha banga important chile hai bacteria propiani bacteria sermani jaga kha they have large holes bangsaga large holes ne wa chile again due to the production of carbon dioxide ke hon escape bukchin hon por kha jang kha omna na por kha na kha bak tep dan mo hoi bang pe kha large holes next Roquefort cheese kichi hai chu atang pia kha banga fungi zanga mo Roquefort penicillium hai zat le kha they will give what ripened flavor nam tui hiu ma cheese te chu nam tui ma lo mo ripened flavor kha ki pia atwa that distinct ripened flavor pen kha hai sia ka hi mo so these are the microbes which are used in household products. Next topic what are the microbes used in industry atam jo sema mo ton in swincho wa hilowa gam sunga dinga tampi khat bol somta lechin we have to use industrial processes right so that industrial production this is number 2 industrial products are we are going to use, which are valuable, which are useful for the human beings. We mostly use we produce beverages more, like all various types more, and then we also use antibiotics, which are effective against bacterial infections more. Beverages and masaniole. Beverages so hai te khabang chi ki bola chile, they use big fermenter vessels more. Vessels lian pisang unchin hoi vessels de kha mam ki chi fermenters. Hai sunga kha apoi mo things ko lu dunchin beverages hile wine bang hile grape nazat ut pen grape tu mo ahilo le ram khong hilo isko khong ile wife flour te khong mo bang khata e bajra chi kho mga hai products of the cereal products de kha nazat pen pen kisang abana tu to nazat de ten te mix combination high vessels you put inside these fermenters, and the process will begin, and we'll get beverages as well as antibiotics. So first we'll see the beverages. Beverages are what are the fermented beverages. Fermented beverages are wine, beer, wine and beer, about whiskey, brandy chui kam la khol ki sa kholom lom aram the whiskey chui design wine beer. So hai de kha beverages de kha so we use yeast tol ki chi ma tol ki zanga ki bol chin hoi tol pen kha banga what is it we call it, brewers and so ki brewer cha ma brewers yeast fungi, it is a fungi, microbes ka bang ki zang fungi. Brewer yeast ki zang chin hoi pe kha malted cereals and fruit juices bowl na mo, so that is used here in industrial production. Saccharomyces cerevisiae. Tama Saccharomyces cerevisiae. But hey, industrial production na wine beer thong hi changa ha brewers yeast kitchen. When it is used in the bakery or in the food products household, we call it what? Baker's yeast. Clear? All right. So wine and beer chew without distillation, aki bol engine. Without distillation. Hai chu ki filtrate si lo process pin ha without distillation hai chu you with distillation. So new khat a process ki ballo jaga kha bang khadan alcohol percentage dile new khat ki baklo ma hai te kha ha lo de hai te kha ha de. So distillation process ki bol jaga kha ki filtrate jaga kha. So chemistry legacy more distillation te kha. So that is how. Number 2 antibiotics kachima. So that is how we use in fermented beverages. We use a fungi known as Saccharomyces cerevisiae. In this case, we call it brewer's yeast. In baking industry, we call it baker's yeast. Antibiotic. Mala. Antibiotic ha. What's antibiotic ha? Bangai sile penicillium here. Quine mumasapien Alexander Fleming. So antibiotics they are used against bacterial infections. So I have accidentally once he was working on a bacteria. All right, once he was working on a bacteria known as Staphylococcus. Hey bacteria nasib. Actually, ha soap sang khalo in paisan khamo sin khalo ta. Actually, hai Staphylococcus bin khai chao. Hai kha a growth kha they cannot grow anymore further. Okay, they didn't grow anymore further when he comes in the next day. Actually, bang ziaga actually kha ele kha ya kha penicillium an om jo fungi mo penicillium an om chin. Data. Hazaga kha agror kha kistobahi chilecho bacteria thathe kha penicillium hi cho mukhya cho accidentally on discover male. So that is how we discovered the first antibiotic. bacteria na sem la ya kha ken khat te chu tham phau mo lucky til khat jackson na bang khat discover un chin ja min thang ma yom mo for that obviously they are working hard only they are working on something mo ile kha ham phadia ko mu chi man thu hi mo aba na penicillin note tam se ga han chit amol ping pin kha so later kha XII leuktin Arnis chain and Howard Florey will use it later to the full potential and also I use this kha why aki discovered to apalakha antibiotic it was used to treat American soldiers infections aparamo World War II and also because of this Fleming chain and Florey were awarded Nobel Prize in 1945 all right other antibiotics were also purified again later tampi ki bol kha to chu penicillium chu ho ilo lampang hita adang aho izo sem tampi ki bol kha kin chin pakat mi hing tia dinga kha ato am to ma kha to treat disease sama kisa to treat deadly disease to treat deadly disease like what plague chi khong ma gama boina whooping cough chi khong diphtheria now ngak the vaccine appear cha DPT ki chi voi ma la vaccine so that I khu mo sao te khong nei lo na diwa khama and leprosy uses jo aya so antibiotic le cha kha kisam to kha next kha microbes in some microbes which are used in for human beings microbes which help human beings we can divide into 3 categories thana kha chita ko khap ne le why they khambanga chile chemicals are chemicals they can form all right they can form chemicals they can form enzymes and also they can form bioactive molecules chi thoba to ke jam tun chemicals elle whatever tun vinegar chi khong anhon na me khong ale ki so voi wala sa khong tom hoite kha citric acid chi khong acid tom ne umale ethyl food ingredients kha we use lots of acids all right a restaurant hong kong ten kha thei diam du ata tom salad khong so ding soup khong so ding chi ne kha to enhance the flavor male so all those they are chemicals all right and they are taken from microbes and those chemicals for example if we write down bacteria can khat citric acid ba ko ya para kilai acid mo e pe citric acid aspergillus niger pa kila fungi aspergillus niger he kha fungus se chu Njin abana citric acid lo le acetic acid le ko para kilai chile acetobacter male simple na acetobacter abana butyric acid chi khong le ma butyric acid le ko para kila clostridium yeah clostridium butylicum you can write it down butylicum butylicum so lactic acid la idan la mai gen zo pen mala lactic acid will be what lactobacillus apat mala so lactic acid la lactobacillus so chemicals were formed were produced from microbes, alright, fungi and bacteria. Some are from fungi and some are from bacteria to produce, and these are used in everyday food items. What about enzymes? Tu napuan nun it yang sarf sobita sarf excel chi khong hilo le bang mga kizang. Ay tiya khap napuan ka siang zele chumale Mr. White chi khong. Actually bang jidan do siang ay chile oil stains how does that get removed? You know, how jidan do mangthaya chile kha enzyme so me chi detergent samo, and that enzyme is known as lipase to remove oily stains. Cloth sama. Here kon pan piu hele mole ni tengai zan mole lipase an achile tun bottled fruit juices je and legit tui juices when you buy them, mango juice, this lychee, so here they were still banga aha chide ya mole to high home mang hele mango juice bang aha bang den o me chu mole here khate chu siang kil o me chu mo, so asyang kil te kha tang pia kha mangza ka siang kil wai chile fruit juices te ho again abang carbonated drinks te hilawa fruit juices te ha mango hela strawberry hela orange hela. So if you see real chikhong mole real juice chikhong le le siang ngai chu mole ai te kha bang za gai chile pectinase ya. So pectinase is used as a clarifying agent in fruit bottle juices. All right, what is it used for? Pectinase to clarify the bottled juices. Pectinase and protease. Pectinase and protease je banga kizang to clarify the bottled fruit juices. Abana khatleom lion chin streptokinase ila ngai tamo ki that thei ta lo chio. Streptokinase enzyme. Streptokinase enzyme lekha ko apara kilai chile streptococcus sapada from streptococcus. Streptococcus sapada kila banga kizangai chile hai lekha clot buster akizang banga clot bad clots pala nasi sanwa blood te kha clotting om ngi le chu mga si san kha khalin chin pai swak thelo janga kha when there is no smooth flow of the blood more inside your body, then you can have heart attacks. So it is used for blood buster in patients who undergo heart attack. Heart attack nahi tia dinga. Paikha myocardial infarction like it's myo chu heart min hilong ma heart lam to kisai no sunga myocardial in FR hilong FARCTIOL. Streptokinase kisam. I think we are done with the enzymes mo. So enzymes, don't forget pectinase to clarify the bottle juices, streptokinase uses a clot buster, and lipases used to remove oily stains. To bioactive molecules. Bioactive molecules are cyclosporin A. Kizang to organ transplant it is used as immunosuppressive agent. Mangchina immunosuppressive agent. Immunosuppressive agent. To organ transplant on by kidney jikhong kha, liver jikhong. Organ transplant janga kha. So you know that they are foreign, right? Those blood tests hong bang hong mga kinai relatives hong achu hithau mo hilele kha atuam heju. Tuam foreign eta chu nang organ adia kha nang body adia kha. So in your body immune system kha we have WBCs kha tuam tuam min chu mai kha antibodies produce we chu. Antibodies chen kha mga apola mapa rahon ki pante antigens ki jiwai te kha fight zelu so that our immune system could be strong and then can be protected from various kinds of foreign materials or diseases. Why antibodies then kha de ilo nang kwai ti to amna cha mo ke body am ngei lo hite, so they'll try to reject it. All right, they'll try to reject, and then the transplantation of that organ can fail. Why saga kha transplantation zo cha doctor ten kha dam doi pizel diu wa immunosuppressive agent to suppress the immune system, okay, for some time, so that the patient will be slowly recovering, and then so that the kidney that is transplanted to him will be working fine, function fine. All right, so why is there immunosuppressive agent ki pezel hai khamau chu nanga neu khat ela banga reactions chu ombe themo neu khat chu athwa ngai chu so pi da mule chu neu khat chu ki thwa na zel ngai mo, and then suppressive agent na ki jang pen khabanga cyclosporin a where organ transplant mo. Ziak chu bang ziak chita antibodies tin kha hey organ adang pen kha ana de ilo dio ana tha som dio ma ana welcome lo wai kha suppress adinga kha cyclosporin a kisa, so antibodies in kha antigens te kha ana tha som mai chu antigens changa chu foreign material hon pai him bim moi istak sa sunga abana alas na bioactive molecules alas na statins kichi statins che bangun cha kizam mo hospital bangun cha kizam statins che ekha banga para ki produce fungi apada yeast apada fungi apada, it is produced from Monascus purus. Statins de banga kizang hai jo organ transplant che ma SAXS immunosuppressive agent. Statins, they are used as blood cholesterol lowering agents. Bang blood cholesterol lowering agents. The blood waha cholesterol omcha cholesterol khali ko apana ki produce tale liver male liver akha cholesterol opta wai kha na bloodstream hon paita cha tam pi hon paita na kha fats hawo ta ne chang ha produce ta chu bang banga tel junk foods ko ma hilo le tel athau khong tam ne khata cha ba da bloodstream ban lo ta ho chang kha ke pakhan na bloodstream kha apai banga ki blood pen kha smooth de kha pai thenon taloma wai pen kha wai hun chang kha bang chi kha banga cholesterol kha level kha sa ngai chu male thau khong ha tam ne le thil kar khong ha tam ne le kha so the blood cholesterol kha hun sang ta mo bang banga gen HDL ha bang banga tom wai cholesterol agent se hun sang gop tai chang kha, so which is not good male dam thailo na dia kha ki dam thailo ta why zaga high cholesterol inhibit thading kha we use statins. All right, so we call it is a blood cholesterol lowering agent. Si sana pa ngi banga kha pai theno lo ja chu bloodstream kha cholesterol tam go ma wai kha hot lo. Bioactive molecules zota to chu atang pi khat ki zo win chin abana next sabang ta ka chi sewage treatment ki ma sewage treatment na bang chi banga microbes de zanga kha inin teo kha ituinin teo neu khat be ki su sang thei diam rivers ko oceans ko nga alut masangwa wai pen kha microbes in sewage treatment that will be taken in the next class. Thank you. Welcome back, everyone. Continuation of chapter 10. microbes chen human welfare akha to akha sewage treatment bangchidan there was sewage. How can we treat sewage? That is a dirty water into a little bit cleaner water before discharging into rivers or oceans. We are going to use microbes. All right, can be bacteria, it can be fungi, more together. More like we are going to use microbes. So what is sewage? Sewage pen kha municipal wastewater. Kichimo wastewater kha igamwa yom namunwa wastewater pen kha municipal wastewater. Kichi hai kha sewage. Municipal wastewater hai kha wastewater. We are going to use, tanks. All right, inside tanks, we're going to use microbes, right? And then in 2-3-step process, the water will be clearer more, and then it will be discharged into where? Rivers. Hey, a process, come on. In municipal wastewater, tanks there we call it white tanks there. Tanks like it is at give sewage treatment plant a plant chile sing hiloma awam namun ha sewage sewage treatment plants zanga ma sewage treatment plants kha a bomb lian pi mo tampi kha do min chin hoi kha as a whole hoi sunga kha tuite kha kinon lo zel jin abana apol ba ki discharge cell hoi plants te kha om namun pen kha mo hai cha in dan de mo plants tampi om ji hoi te kha ki go through second chin and then it is discharged to rivers or apart min kha where the sewage is treated. Okay, the place where sewage is treated is known as sewage treatment plants. We're going to study the in-step process. More step thum kong mention why te kha yen de wala. Banga aim kha to make the water less polluting. What is the aim of sewage treatment? By using sewage treatment plants, so that we can treat the municipal wastewater and make it less polluting, because the municipal wastewater is so polluted. There will be pollutants, there will be soils, pebbles, dirty water from all the household, right? So that is why we are going to treat them. The first treatment is known as primary treatment, and the second is known as secondary treatment. Primary amasazo. Primary treatment, mostly you're going to do the physical work, the taking out of the physical debris, removal of the physical debris, the bottles, more the dirty clothes, polythenes, more lian physically you are going to take it out, and then there is the filtration process, and whatever is remaining behind it will go down to the sediment, and that is known as sedimentation. Primary treatment will have filtration, removal of the physical removal of the debris, and then whatever is remaining, call it sediment. The process is sedimentation. Primary treatment, we have 2 steps: filtration and sedimentation. So, filtration and sedimentation. Tunantale yaita. Tuita. Bottle home, polythene home kila khet bektacho. So, hai kha anwaya kha. Om te kha, hai kha sediments te kha. Sedimentation ama, that sediment we call it primary / sludge. LUDZE. Primary sludge. Mga primary sludge. All the solids that settle, okay, that forms the solid dekha omlai dekha primary sludge kichi mo, and then to primary sludge bir kha annex kha ki puiding engine next thing mole next thing ki puitau di wai kha bang kichi di secondary treatment. Next will be what? Secondary treatment. All right. Oh yeah. Secondary treatment. Primary sludge ma. Primary sludge solids aki aloya ki settle down ti khas second khas a secondary treatment ki puidi. Secondary treatment is also known as biological treatment. Why? Because we're going to use living organisms. Biological, we're going to use living organisms. So primary sludge ben khatwa kha bang bol tadi. Air mala. Air kha ki pump tadi. And it will contain aerobic microbes. Yeah. Bacterial mala. And then the water here, the primary sludge, they will form a mesh-like structure, which is bacteria plus fungi. Why mesh-like structure pincha MES kichi. It will form a mesh-like structure known as flux. Flux, important. Very important. What are flux? Why have mesh-like structure? So what are flux? Flux are masses of bacteria associated with fungal filaments to form a mesh-like structure. And how is the secondary treatment started? They'll aerobic microbes. They'll form a mesh-like structure known as flux. And then after that, for them it is food, right? For them it is the organic matter. For them it is the organic matter, it is the food for the microbes. So they eat it, they digest it. So while digesting, in the process the water will become clearer, and then we can test it. There is one term known as BOD, biological oxygen demand. We are going to test it if the BDO is still high or low. So BOD is biological oxygen demand. How much these microbes, they eat the organic matter and oxidize it in such a way that the water is becoming clearer. So BOD, if it is more, still more, then it is more polluted. All right. Concept ka. If BOD, if it measures less, then it is less polluted, the water. So it is a measure. To secondary treatment, what do you think it will be? The BOD will be more or less, because the microbes, they use the food, the organic matter, and they oxidize it. So the oxygen level will be a little bit more higher. If the oxygen level is a little higher, that means the water will be less polluting. That means it will be more clearer. So that means what the BOD will be 100% lesser. So it will be less polluting. So secondary treatment BOD is less. That means what water is less polluted. So what is BOD again? It's a biological oxygen demand. All right. It shows the amount of oxygen which is consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. Oxygen level water will be less polluted. So that is how we measure it. All right. So then it is now better, less polluting. So whatever is present at the bottom again, bottom present sediments, so that when the BOD is reduced, all right, BOD reduced here, BOD is reduced, and after BOD is reduced significantly, then that the sediment, now here we call it activated sludge, primary sludge to activated sludge differences, important more, SLUDCE, sediments ka, which are present down mala or activated sludge kichi, okay, difference with primary sludge while lekhanigal thayuma activated sludge kichita. Activated sludge pirkha, if you want it, you can use it as inoculum starter mala in the first thing mala aeration thing kichi, aeration thing kichi jang achubang air ki pump na thing pin kha then lama ka drop in kha waya kha start ki thading chin the process you can start again ma if you want it. So aeration thing kha aeration thing ba kitale mala air ki pump pin kha thei lai diwa ma then lama air pump ka chi pin flux ki form na pin kha ma aeration thing akha, you can start it as an inoculum starter and then start again the process. So this the sediment in the secondary treatment, the sediment which is formed at the bottom, that is known as activated sludge. About the next akha, now why water pure BOD it is reduced. So now what happens is that you can go in two ways. What are those two ways? Next step. Activated sludge, acylated sludge, inoculum azate, more, and start the process once again. Number two, you can start another process. Then next ten khada by sakki dichina. I will write it here. Pumpkin anaerobic sludge digester kichi pangzak anaerobic oxygen tel non loma hoido ke sin kipta sealed. So anaerobic microbes will start, anaerobic microbes male, they will start digesting whatever is inside. Anaerobic sludge digester kitchen, anaerobic microbes will start digesting it, and then they will form what? Gas ka, form the biogas kitchen. Biogas le form thema. Why biogas bekha is adule food, cooking food konga le kizak mala methane, carbon dioxide kha tel cha. The cocktail engine is used for cooking food. Gas on the way jumala. Why like is first? To high borus is low lele at which they has young to him that water is already, the BOD is already reduced. So now what you can do is that you can release it to the reverse. It will be less polluting. That is how we treat sewage. Clear? So who is taking part? Whose initiative? And so we'll have rough take mug. Now that is how we treat the sewage. Ganga XII plant and Yamuna XII plant kichom inchin Ganga to Yamuna ha mole India dinga ahoy dinga reversely and pen here Ganga Yamuna mohati honga sewage treatment plant ki ball inchin. So that when it goes into the rivers, it will be less polluting. We have different programs. Ganga XII plant kichihoma government take up. So that is how we treat the sewage. A second tun biogas to bolu talachin. Tui to kisai hello. Now it's not about to make the water less polluting. It is just about if you just want to produce biogas. Village. You can put here bit khat bol na, let's say 10 to 15 feet. You can put cow dang water yeah you can seal it mole hele anaerobic microbes tin khana sem di chu and then the gas will be formed which is known as biogas mole methane carbon dioxide mo gobar gas lekichi bonga pata ki bole changa mo gobar gas lekichi same thing and then methane to carbon dioxide so this uses efficient cooking fuel in villages mala so if you just want to do that you can also do this gobar gas now the question is how can a cow dung produce methane all right methane CH4 how can the cow dung produce biogas mala because the reason is and to tell more biogas produced at 10 to 15 feet cow dung water slurry mixed anaerobic microbes will start working digest or dela maigen dan to mo anaerobic sludge digesters to kiba and then biogas will be formed actually kawin bang chinan do akha methane biogas produced here mi chile akaw dang akha bacteria mai chu why bacteria min khabang agilwa mo arumin wakha they have a bacteria known as methanogens group of bacteria which can form methane they can form methane why amin bangai chile methanobacterium hai ne we chu hai ne jang wa kha kawin kha rumen part of the stomach kha methanobacterium name chu hai kha group of methanogens kha methanobacterium kha example hain kha akhai kha kaw ben kha kaw dang hon ne chang kha ekma bong ek ne chang ha kaw dang kha bangom lai bacteria ti om lai chu mole so that is when you put in the pit mole so that is how we form biogas methane carbon dioxide mole here that is how we get it we can get what biogas and biogas contain what CH4 carbon dioxide ma hydrogen sulfide chilium like so why think kha ki form the kautin kha elo chin hey khong ne we chu mala pol khong ai pol te kha digest ne ting wa kha mo cellulose pen kha very hard chu why digest ne ting kha e methanobacterium nei lo le mo mau digest thei lo di why digest thei de wa an bong chu an channel jin ba na hai theu ma le ola gella ma ki digest pan hi lo ma le cha hai theu ma le san khwa khwa le ja chu ma kha e cha banga regurgitation process dan de wa mo la khat ki theu chu hai theu chin but apoimo pen chu the main point is that they have a group of methanogens like methanobacterium okay which contains methane so when it comes out in the cow dung and when we used it and seal it the pit then we can form biogas that contains methane and carbon dioxide right that is known as the production of biogas so the question what is biogas mo or gobar gas banga what are the main constituents of biogas theo ya mala kha cooking fuel la kha hin chin cow dung apa da kilamo that is produced from the cow dung this one is the biogas it contains methane and carbon dioxide clear actually idella ma question kha banga what are the two steps in sewage treatment sewage treatment we have two primary and secondary treatment so water flux mesh-like structure consisting of bacteria and fungi we have flux actually what is the difference between I did don't tell you to get collector difference between primary treatment a sediment ka primary sludge and activated sludge banga difference. Activated sludge ka secondary treatment na sediments de ka here. Other flux. What is the difference between primary sludge and activated sludge? Clear? To next step, we are going to use microbes as next abang ata biocontrol agents mo. Bio to living yamala. We are going to control the insect and pests using the living organisms, that is microbes. Mala bacteria, fungi, virus, we are going to control the insects. How is that possible? For example, if you want to control mosquitoes, then what can you use as a biocontrol agents? You can use ladybird, dragonflies, mo tweet he khongle mauha net he weju ne kuchia kha alarvi de khongle mosquitoes de khongle mauha net he unchin hoi kha it will control the mosquitoes. So it is acting as a biocontrol agents. Are they living or dead? They are living. So ladybird, dragonflies, in order to get rid of aphids and mosquitoes. Aphids they were what is small insects which can suck the shape of the plants. Sucking insects. So that is how we use the biocontrol agents, alright, to control the mosquitoes and aphids, ladybird and dragonflies. Next, very very important, second pin khabang. BT, Bacillus bacteria, Bacillus thuringiensis, spelling ka sao mo ay ka thuringiensis, thuringiensis, GIENSIS. Yain mang tiyan do pan pili ko ya bang plan na to ka let's say cotton plant mo cotton ka abang kat na disease pin ka video mention ho itin ka mau ka disease sakzelo mo na sakzelo ja cotton production ka nyam gi cho. So scientists were thinking what bang bol tawai jile bacteria zanga hai Bacillus thuringiensis in ka cotton bollworms te ko mo worms se chumala bang chidan de wat hadde living organisms zanga Bacillus thuringiensis zanga mo. Genetic engineering biotechnology you're going to study the whole story about BT cotton mo like BT, the relationship between BT, Bacillus thuringiensis, which is used in cotton so that we can get rid of the worms. Alright, so how does it work? BT toxin it has toxin. Okay, toxin like larvae sunga bollworm larvae sunga chitani bollworm larvae. Inside the bollworm larvae, when we spray the BT bacteria you can buy in the form of it is salt in dry packets. Okay, like spray the issue. Alright, spray water to mix the spray inside this bollworm. What happens is that they will release their toxin, the bacteria will release their toxin, but when they have the toxin, alright, they have a deadly toxin inside them, but it will not kill them. They can survive in their own in their own pH level ki banglaw alkalinity pH level ki banglaw hayon tung chang bollworm spray chang when we spray the bollworm inside the gata they will release the toxin. Okay, toxin release the they will die. Yeah, so that is how we used BT as a biocontrol agent. Alright, engine hoi kha BT, the cotton which is resistant to the bollworm. Now we call it what? BT cotton. Hoi the disease of no low engine high spray chang khana we are banga problem of no low more. You can produce as much as cotton in your field and you can sell more without any disease cotton plant. So insect pest ya keep preventing ama BT kizang. Adana kon biocontrol ha adang le tampi kizang engine tam mo wai kachi jaga a species minte khamo na thei ngai chu. Trichoderma le kizang hele fungi. Hele kizang trichoderma mo hele kha ko atampi ha ruta mo root pathogen which can cause disease mala an organism which can cause disease why they root system root ecosystem trichoderma le kiza adanga viruses entally virus abang kizang baculoviruses le hoi biocontrol abang chidan de baculoviruses bakchidan de wakizangai chile arthropods mo exoskeleton ne wai millipedes comb crabs all those arthropods we can kill them by using what? Baculoviruses. Alright, that is how they are acting as a biocontrol agents. And then baculoviruses were coming from a group of genus known as nucleopolyhedrovirus. It's a genus, heterogeneous. Baculoviruses, they are very effective against what? Against what? Arthropods. Wow. So this is the last one. So all these, if you want to, reduce the population of all these pathogens which can cause disease in plants, you can use biocontrol agents. And this is done overall under integrated pest management. Government aparama integrated pest management waya kibo program ecological sensitive sing te lo de na poimo protect ngayom te dia ka kha wai te kha hai te kisang so that we can protect the ecologically sensitive areas. Next microbes as biofertilizers. Why do we use? There are some living organisms which can act as a fertilizer, okay? Nang chemicals dia sang selawa mo to fix nitrogen. Mala. Why we are going to use biofertilizers in some plants in paddy fields, and we need to know what are the name of those bacteria. Here, why blue-green algae, so we can use them to fix nitrogen in the soil. It can be bacteria, it can be fungi. Here, microbes microbes so bacteria tampia banga kisang. Biofertilizers organic farming that's organic chemicals organic farming farmers bang siang thoya organic vegetables banga kilengu dia bang ma har bang ma lo chemicals bang ma kisang lo siang tho anam liom lo siang tho chen isunga dia le hoi why the organic farming kitchen why the biofertilizers kisang paddy field bacteria le kisang fungi le kisang fungi le bacteria pen banga rice, for example, rice banga example ichile nitrogen fixed heading azospirillum. Mintam tamo by hardio azospirillum, azotobacter, phylogeny cyanobacteria, blue-green algae. Here, example bang anabena nostoc, ana nostoc, oscillatoria, nostoc, oscillatoria. Hai ti chukiza. Cyanobacteria and nostoc, oscillatoria, these are the bacteria. How can they help, as a biofertilizer? Because they help to fix nitrogen in the soil, in the paddy fields. That is how it helps. So, actually, last in fungi, we have rhizobium. Class 11 na leon male. Kaya roots male. Roots of leguminous plants. Leguminous plants. Here, pea plant itacho. For example, pea. Kaya rhizobium. Here, they associate with the roots. They live together. The roots will give them shelter, and they will fix nitrogen for the plant. For the plant, rhizobium. Roots of leguminous symbiotic association. Here, see the roots and the rhizobium. These are rhizobium. So this association is known as what? Symbiotic. They cannot live without each other. Symbiotic association. Symbiotic association plana on chile rhizobium and roots of leguminous plants. So this is just how it helps as a biofertilizer, the microbes, the bacteria and the fungi. So currently in our country, a number of biofertilizers are available commercially in the market, and the farmers use this regularly in the fields to replenish the soil and nutrients and to reduce the dependence on chemical fertilizers.